In this screencast, you're going to learn all about probability plots. In exploring the nature of a set of data, an early question arises as to whether the data appear to have come from a population that is described by a particular distribution. For example, is the normal distribution appropriate for your data? Or is some other model more appropriate, like the gamma distribution, or the exponential distribution, or all of these other distributions that we've talked about? Which distribution does my data likely fit? In this class, in the last couple weeks, we've already seen a couple examples of how we can fit data to a distribution. Here's an example where we had 1,000 different measurements collected and we fit the data to a nice gamma distribution. We also did something similar with the male and female heights in the class. We made a histogram. But as you see, as the number of samples starts to diminish, here we have 48 female measurements and 58 males, the number of bins that you use in a histogram starts to decrease. And so as n decreases, you can't really see the distribution as much, and you don't really know, you're not as confident as to which distribution your data follows. Today we're going to be talking about some measurements of brown trout in the Grand Canyon. And here I've just created a quick histogram, and we only have 10 measurements here. Sometimes your sample size is quite limited. And I just have a histogram here. With only 10 samples, there's no way that you can make any sort of inferences on the type of distribution simply because you only have 3 bins here. So for large samples, a histogram can be used to fit various distributions with ease, but for smaller samples, this becomes quite difficult. Although there are different types of quantitative calculation methods for addressing this question, the typical approach is to use something known as a probability plot. So today I'm going to explain what a probability plot is, how you can generate them, and then the easiest way to generate them is in a software tool such as Minitab. So let's look at some data. We have 10 samples, the length of brown trout in the Grand Canyon. I've got a friend who is an ichthyologist, that's a fish scientist in the Grand Canyon, and he basically just goes around and collects fish and does studies on them. He sent me some data. This is the observed length in millimeters. We can compute some sample statistics, the average and the standard deviation. And in order to figure out what type of distribution your data follows, you have to start out with a hypothesis. So we're going to start with the hypothesis that the length of brown trout is a normally distributed variable. And then we're going to see how well these data fit a normally distributed variable. If this is true, so if the data follow a normal distribution, then our probability density function should be the normal distribution, centered about 152.6, with a standard deviation of 24.9, as shown here in this plot. Now, if we were to take 10 samples from this population that are characterized by this distribution, the question arises approximately where should they lie? And you guys all know, because of how these probability density functions work, if I had 10 samples, each of these regions here, separated by the dotted lines, represents a probability of 10%. So this first region here is 10%. This second region here is 10%, and so on. If we have 10 samples, we would then expect to see one fish in each of these 10 different categories. If we calculate the cumulative probabilities of each of these red lines, now note that I only have 9 red lines because we're splitting this into 10 equal area sections. So I've got the cumulative probability of all the dotted lines. In fact, if I wanted to put 1.0, that basically doesn't exist because it's infinite. You would have to go all the way down this curve on the normal distribution to get exactly 100% by definition. So these are the approximate x values that are corresponding to those red lines. If we plotted this on a cumulative frequency diagram, capital F of x, we can see kind of the same sort of thing where we have equal increments going on the y-axis here, and these are the x values corresponding to that. Now let's revisit the fact that we can't have 1.0 because that's really infinite. What's typical is to sort of think of these different increments on the y-axis as bins, and we sort of want to take the bin center. So we're going to look at these red markings here. For us, that would be 0.95, 0.85, 0.75, and so on. That's what we actually do is we sort of back off and we calculate the centers of those quote-unquote bins, represented here by these blue lines. And from those, we can then calculate adjusted x values that correspond to our predicted lengths in millimeters. And these are known as the expected cumulative probabilities. Again, instead of using 0.1, 0.2, and so on, we back off and we kind of do a bin center. And that means we get 0.05 all the way up to 0.95. Using the norm.dist function, we can calculate these x values in Excel. How do we calculate these expected cumulative probabilities in general? The cumulative probability expected is going to be I minus 0.5. I is just the index going from 1 up to n. And then we divide that quantity by n. We saw that when n equals 10, we get the following. But for example, if we had n equals 23, then the expected cumulative probability would be as shown here. And these are just calculated from this simple formula over here in the middle. The old way of doing these probability plots was on something known as probability paper. So you could actually buy these pads of probability paper. And this is just showing one example. The y-axis over here is not linear. It's almost like a log plot, but it actually goes up and then comes back down. So the top and bottom of the y-scale here has very small increments, and then they increase towards the middle and then decrease as you get down to the bottom. The old way of doing this would be to first sort your lengths. So we would first have to sort the lengths in ascending order, and then you would plot these on probability paper. So over here on the right, I have sorted our lengths from smallest to largest, and those correspond to our cumulative probabilities, 0.05 up to 0.95. On normal probability paper, we would then plot these pairs. For example, we have 105 that occurs at about 0.05 frequency. We have 134 that corresponds to 0.15, and then we can keep going. Now, on probability paper, if your data appear to be linear, like they do here, the conclusion that can be made is that the data, these 10 points that we have, are normally distributed. All right, so it's a somewhat qualitative method where you just kind of look to see if these data are linear on normal probability paper. There's also exponential probability paper. There's Weibull probability paper. There's all sorts of probability paper for different distributions. If we plotted our data on exponential probability paper, you can see that it might look a little linear in the middle here. In general, it's not very linear because we have this point down here and this point up here. So this appears to be very nonlinear on exponential probability paper. The conclusion that can be made here is that these data do not follow an exponential distribution.